sugar. Oh, honey, honey.
Hi, I'm Joy Galloway, a graduate student of nutrition science at the University of Hawaii at Manoa, here today to talk to you about nutritional biochem and carbohydrates. Carbohydrates are the macronutrients from our diet that we rely most on for energy. Carbohydrates are also called saccharides and are commonly classified as being either simple or complex, with simple being monosaccharides and disaccharides, and complex being oligo and polysaccharides. I'm sorry, did you have a question? Saccharide units are 6-carbon aldehydes or ketones that are isomers of C6H12O6, the most common being glucose. Carbohydrates are called such because they are hydrated carbons. The main 3 monosaccharides that we consume are glucose, galactose, and fructose. However, we do not consume the molecules as individual saccharides, but generally as disaccharides and polysaccharides. The 3 disaccharides are sucrose, maltose, and lactose. Polysaccharides are long glucose chains like starches or dietary fibers like cellulose. Starches may be an unbranched chain called amylose, or branched chains called amylopectins. Now, dietary fibers have various structures, but because they have different bonds than the starches that humans cannot break, they don't provide energy from glucose and aren't detailed in this video. Most of the carbohydrates that we consume are polysaccharides. Starches, which are commonly found in grains, fruits, and vegetables, are rich sources of complex carbohydrates in our American diets and are an important caloric source globally. Other common sources of carbohydrates are dairy products, sucrose, and products containing high fructose corn syrup, and are becoming increasingly common in American diets and apparently canine diets as well. Get emoji. Go on, excuse me. Good girl, Mojo. In order to release energy, carbohydrates must be converted into glucose or its glycolysis intermediates. In this video, I will explain how dietary carbohydrates are digested, absorbed, used for energy, and converted into storage forms for later use. Let's talk about how carbohydrates get digested in our body. To appreciate digestion, you need to understand the critical role that enzymes play in causing carbohydrate molecules to be broken down ultimately into monosaccharides in the digestive tract. The enzymes involved in carbohydrate digestion are hydrolytic enzymes. Hydrolyzation occurs by water being added to a substance to break bonds. For hydrolyzation of disaccharides and polysaccharides to occur, the activation energy of the reaction must be decreased. Digestive enzymes, carbohydrases, allow this to happen. The first enzyme that ingested carbohydrate encounters is salivary amylase, or more exactly, salivary alpha amylase. Salivary alpha amylase is produced by the salivary glands and is part of saliva. Alpha amylase hydrolyzes the alpha-1,4 linkages of starches, the unbranched portions, resulting in the disaccharide maltose, or shorter poly or oligosaccharides. Alpha amylase cannot break the alpha-1,6 bonds that make the branches of amylopectins. Once the food has been chewed and has had amylase acted upon it, the mashed food is swallowed and is now called a bolus. It passes through the esophagus and enters the stomach. In the stomach, the very low pH destroys the enzymatic activity of alpha amylase. Here, only mechanical digestion takes place. That is, the strong muscles and gastric juices help to break apart a bolus until it is liquefied. The substance is now called chyme. Chyme enters the small intestine slowly, about just 1 teaspoon per minute, through the pyloric sphincter. The small intestine is the main site of carbohydrate digestion and absorption. As the contents of the small intestine move along by peristalsis, alpha amylase again works to break the alpha-1,4 bonds of the unbranched portions of starch chains. This alpha amylase is secreted by the pancreas into the duodenum through the common bile duct. To digest branched starches, the alpha-1,6 bonds must be cleaved by the enzyme isomaltase. This enzyme is produced by the mucosal cells of the small intestine itself and is found on the microvilli, acting upon 1,6 bonds that come into contact with it. Also on this brush border are the hydrolytic enzymes that cleave disaccharides, named by which molecule they cleave: maltase, sucrase, and lactase. Now that the carbohydrates are free monosaccharides, they are ready to be absorbed. The small intestine is very efficient at absorbing monosaccharides. Glucose and galactose are absorbed by active transport, but fructose is absorbed by facilitated diffusion. For glucose and galactose, a protein complex uses ATP to bring both sodium and glucose or galactose into the mucosal cell in exchange for potassium. Once in the epithelial cell, most of the glucose enters the circulation by a transport carrier called GLUT2, though some glucose simply diffuses into circulation. Fructose, on the other hand, is not dependent on ATP for absorption, but uses a transport carrier called GLUT5 that moves fructose into the cell via a concentration gradient. Though it is more energy conservative than glucose and galactose, absorption of fructose is much slower. Fructose is transported out of the mucosal cell by GLUT2 into the capillaries. The primary uses for carbohydrates are for energy production through glycolysis, the Krebs cycle, and the electron transport chain, and for storage of energy in the form of glucose chains as glycogen or conversion to fat as adipose tissue. Once carbohydrates are in the bloodstream as monosaccharides, they are carried directly to the liver. In the liver, the fate of the monosaccharide is determined by whether the body is in need of energy or not. This determination is communicated by hormones and enzymes. Most glucose is metabolized in the liver, as is virtually all fructose and galactose. But some glucose passes on throughout the body's circulation to be distributed to other cells like muscle cells, brain cells, and fat cells. Passive diffusion through transport proteins moves glucose into these cells to provide energy. Because glucose is such an important energy source in the body, maintenance of a stable amount of glucose in the blood is tightly regulated by hormones. An important hormone for the uptake of glucose in muscle and fat cells is insulin. Insulin is released from the pancreas into the blood in response to increased levels of blood glucose. Insulin stimulates its target cells, such as muscle and fat cells, to move GLUT4 transport receptors to the cell membrane. This allows glucose to be taken into the cells. Without proper amounts of insulin, or when cells are insensitive to it, the hallmark sign of diabetes occurs: high blood sugar, especially after ingesting carbohydrates. After moving into the cytosol of the cell, glucose enters an anaerobic, energy-producing chain of reactions called glycolysis. Through these 6 reactions, enabled by specific enzymes, glucose is converted to 2 molecules roughly half the size of glucose, called pyruvate, and yields 2 molecules of ATP through the breakage of these bonds. Fructose and galactose enter the same glycolytic pathway, but at different entry points. In 3 reactions, galactose is converted to glucose 6-phosphate and proceeds down the glycolytic pathway. Fructose is converted to fructose 6-phosphate or 1-phosphate to continue through glycolysis. Once pyruvate has been formed, it enters the mitochondria of the cell, is decarboxylated, and with coenzyme A produces acetyl-CoA in a dehydrogenase reaction. To continue producing energy, the acetyl-CoA will react with water, oxaloacetate, and the enzyme citrate synthase to enter the tricarboxylic acid cycle, or TCA cycle for short, which may also be called the Krebs cycle. The TCA cycle is a series of reactions that I won't detail, except to address three main points about it. The first point is that 90% of energy from the food we eat comes indirectly from this cycle. The TCA cycle by itself only generates one molecule of ATP per acetyl-CoA molecule, but its energy-producing value is through the electron carriers NADH and FADH2, which results in around 17 ATP molecules in the electron transport chain. And remember, each glucose molecule produces two pyruvate molecules, so the amount of energy linked back to the monosaccharide is double. This is the major energy-producing mechanism called oxidative phosphorylation, which occurs in the inner mitochondrial membrane. In this chain, NADH, FADH2, and oxygen help ultimately create energy as hydrogen ions move through the membrane, driving synthesis of the third phosphate bond in ATP. I've explained how important the TCA cycle is for using carbohydrates for energy. The second point about the cycle is that even though oxygen isn't a reactant in any reaction, oxygen is required for the electron transport chain to function, and if the ETC is inhibited, NAD and FADH cannot be recycled back to keep the TCA cycle running. The third important point about the TCA cycle is that it is not unique to carbohydrate metabolism. Fatty acids and amino acids also may contribute to the TCA cycle, and how this occurs is detailed in my separate videos on those macronutrients. The process of producing glucose from non-carbohydrate sources is called gluconeogenesis. We've talked about what happens to glucose after a meal, but what about in between meals? Cells still need energy for functioning, so our blood glucose needs to be kept constantly replenished. I suppose some of us could just keep eating and eating if given the chance, but we really should rely on breaks between meals. Besides the 5 or so grams of glucose that usually circulate in our blood, glucose is also stored in our liver and in our muscle tissue in highly branched chains similar to amylopectin called glycogen. When there is abundant energy, glycolysis and the TCA cycle are inhibited, and metabolism shifts to anabolic energy storage. The production of glycogen, called glycogenesis, occurs when there is adequate glucose 6-phosphate in the cell, more than is needed for energy conversion. A series of two reactions results in what is called UDP glucose. These molecules are attached to each other in alpha-1,4 bonds with help of the enzyme glycogen synthase. This enzyme is stimulated by insulin and helps reduce free blood glucose following a meal. To make for more efficient storage, a branching enzyme cleaves glycogen chains of 7 molecules and transfers them to another molecule on the primer backbone with the 1,6 bond. Glycogen can store about 100 grams of sugar in the liver and 400 grams in the muscles, for a total of about 2,000 calories of sugar. These storage sites provide glucose for fuel during times when we are using a lot of fuel per minute, such as in exercise, and also during times between meals and when we're sleeping. Tapping into this fuel source is what glycogenolysis does. As its name indicates, glycogenolysis cleaves sugar molecules from the glycogen chain. The enzyme glycogen phosphorylase acts by cleaving molecules with alpha-1,4 bonds, and the enzyme alpha-1,6-glucosidase cleaves the branches with 1,6 bonds. Glycogenolysis is stimulated by the hormone glucagon, and epinephrine and cortisol also stimulate glycogen breakdown, so glucagon and insulin are regulatory hormones with opposite effects. I just don't get it. An easy way to remember which key metabolic hormone is catabolic and which is anabolic is insulin wants glucose to come into the cell and into glycogen, and glucagon wants glucose to be gone. You can see now that not only does the highly branched structure of glycogen make compact storage, but it also makes for simultaneous cleaving of terminal molecules. Well, our body has another storage form for excess carbohydrates: body fat. When glucose enters the cytosol of the cell and is converted to glucose 6-phosphate, if energy is not needed, this molecule may enter a separate anabolic pathway called the hexose monophosphate shunt. This pathway is a series of 3 reactions that generate 2 molecules of NADPH. This NADPH is the critical reducing agent required in fatty acid synthesis. Therefore, excess carbohydrates consumed in a fed state stimulate the production of fat in the body. NADPH is also used for maintaining healthy red blood cells and effective metabolism of pharmaceuticals in the liver. The other important product of the hexose monophosphate shunt are the pentose sugars that make the backbone of DNA. I hope you recognize that carbohydrates are very important. The energy yielded by 1 gram of carbohydrate is 4 calories. The RDA for carbohydrates is 130 grams a day. This is the very minimal amount needed to support brain functioning. The brain is a big user of glucose, and although other tissues are able to use the other macronutrients for energy, glucose is the only fuel the brain accepts. 130 grams of carbohydrates, however, would not be an optimal intake for most people's diets because we need glucose for more than just brain functioning. The acceptable macronutrient distribution range is 45 to 65% of our caloric intake, which, for a person eating 2,000 calories a day, is 225 to 325 grams of carbohydrates. I hope you enjoyed learning about carbohydrates with me. Thank you for watching, and from Mojo and me, a warm aloha.
Sugar.